Ah, there's my red light. The red light says go man, go man, go. So hey, let's go then. Hey, welcome back. Thank you for connecting with us in this way, for continuing your educational journey. And I always say this, man, I enjoy being a part of it. Whatever school we are connected to, whether you're at the high school, man, enjoy those days and the different personalities and the different energy levels and interests that the students have. Uh, just love being around that, and of course, Crowder, man, I love those evenings, hanging out, talking science, talking biology, and being a part of those journeys as well. My eyes itching. There we go. All right, photosynthesis round two. Man, we're going to see some great stuff today, and I got some strong teacher moves. Man, we're going to be. I'm even going to use an animation. Ooh, man, that is some strong teacher stuff right there. We're going to go through that at the end to kind of showcase and heavy review on everything, and just to give you one last look. The only drawback is I got to click through the slide and move it to deal. So, and I'm not real sure. It won't be on the whiteboard, so I won't have the writing stuff. I'm hoping that, let's look. I think there's some stuff in here. Yeah, that might be some drawn stuff right there. So maybe I can still do a little bit as far as that. But we'll see. We'll see how it goes, and it may be a train wreck. It may be a beautiful ride on a sunny day, but we'll see how it goes. But anyway, the idea is to fully comprehend what is happening during the photosynthetic process, because like a lot of this stuff, and I say it over and over again, it's just an amazing process of what's going on. I mean, to think about in the cells of plants. I mean, we mow the grass. I mean, we trim trees. We'll pick up leaves and whatever. And if you're family farms, then you are heavy into plant use and plant production. But man, what's going on at their cellular level is unbelievable. What they're doing and making and producing. And if you just look at a blade of grass or a leaf or a corn plant, you're thinking, nothing going on. But man, there is a bunch of stuff going on. And it's so incredibly helpful to us. It's just an amazing system, as all the systems are. And I just love talking about it. And I'm just in awe of what it's got going on. So, hey, let's get started. Let me get a little box here. And we'll throw in some background information here, because we're going to kind of set up what's happening with some stuff. So let's give this a nice fancy title. Oh, not that one. Photosynthesis process. Yeah, that's a good strong, and some good strong teacher moves right there. Little you know what? I'm going to do it differently. I'm going to do this route, and because we got two things to talk about, and within those two things, they have their own notes. So then we'll use the strong teacher move to identify that they go under that. So when you're writing your notes, it'll make more sense. All right, our first section of the photosynthetic process is the light dependent. And the fingers aren't wanting to work today. Reaction. Capitalize that since everything else is capitalized. All right, and then we will go down, and then we'll space a couple, and then do our little line, just to showcase a little indentation. Requires light. And guess what that light is? Hopefully, ooh, I got two E's there. I don't need that. Hopefully you didn't write down two E's. Requires light, and guess what that light is? Man, I hope your hand shot up, and people were all across this area were screaming at the screen, yeah, the sunlight. No, let's do it differently. I started out wrong, so we'll just do it this way, so you don't have to erase a bunch of stuff. Yeah, capture sunlight and convert it to energy. It was supposed to be capture energy from sunlight, which is the same thing, but I started out wrong, and so I just fixed that, which is a strong teacher move to be able to start it off and then just switch it back to what you were trying to say, even if you didn't start it out that way. And we are going to make ATP and where's the and thing? Oh, there it is. Oh, I lost it there for a second. Now, NADPH. Guess what that is? And you heard a bunch about it in chapter 7. Dramatic pause there. Hopefully you were screaming at the screen. Yeah, that's my electron. That's my electron carrier. That's right. And what's different about this one? Yeah, it's got an extra P on there, an extra phosphate. And that phosphate is going to be used to make the ATP. Okay, so when we see it, it's going to be NADP. Okay, and then that phosphate gets used to make the ATP, so it's going to transfer. So the NADP is going to be oxidized because it's going to be losing electrons, and then the ATP will gain it, so it will be reduced because it's gaining those electrons. So those are words from our past, and of course you got the H there, and the H represents the energy because it is carrying the electrons. And we'll talk about that, but when we look at the animation, it'll really be obvious as to where those H's are coming from and what's going on there. Let's see, is that all I wanted to say there? Yeah. Okay, and now going down, part 2. I'm going to type this whole thing, and then I'm going to go into my spiel because this is going to have a little bit of historical reminiscing, as you know I like to do. Okay, that's the name of the process. It's part 2. It's where the glucose is actually made. So you capture it in the part 1, and you make the glucose in part 2. But now, if you're a high school student, when I was your age, that wasn't the name of the process. No. What do you think it was? If the first one was light-dependent reaction, in this section, spoiler alert, I'll give you a little hint, it doesn't require light. So guess what it was called when I was your age if you're in high school? That's right. Hopefully you were screaming at the screen. Well, it must have been light independent. And that doesn't feel right. Is that right? You know what? I think I did that right. So that's what it formerly was called, just playing off the idea of one needed light and the other does not. So we'll call it light dependent, light independent. But that went away somewhere along the way, and now it's called, with a super fancy title, the carbon fixation reaction. La-ti-da, yeah, that's pretty highfalutin stuff right there. I had an uncle that said that, and I'm not really sure what it means, but I know what he meant when he said it, but I don't really know what the falutin means. But anyway, that's what he said. So call him up, ask him. No, don't do that. You don't want to talk to him. All right, and so then we would go down and a little space, a little strong teacher move, outlined format, and I've already talked about this, but we will officially write it down. Does not require light, and one more, uses ATP, and guess what else? That time I knew where the and sign was, DPH. And that's the end. So I am going to close that out and pull up the old pen here. And a little green. Yeah, we got to go green if we're doing photosynthesis. And put my 2 there. There, as it should be. All right. So that is our pre-look at photosynthesis notes, a little background information. And so what we're going to look at now is just a series of diagrams. And we're going to do more drawing and circling and just talking about it. And when we get to the end, there's a slide, or not a slide, but a diagram. And then we'll type everything out. We'll actually put some notes down. And that's what you will want to capture. And we'll do that. So let's look at everything. This slide, this diagram here is a kind of an overview and what we just talked about. And so if you think about how this relates to this is part 1, and then this is part 2. And man, you can already see this, and we'll talk about it more once we get there. But a cycle, okay? I love me some cycles, you know that. And when we look at this, you're going to say, whoa, 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 whoa. We've seen that. Well, it's going to look really similar, but it is going to be different, because the one we saw in chapter 7 was breaking down the glucose. And we talked a lot about how the glucose would be oxidized and the electron carriers would be reduced. Well, guess what? The roles are going to be reversed this time, because we are building a glucose molecule. So things are going to come in and they're going to drop things off. They're going to be added to the molecule as we try to build the glucose molecule. I say we, the plants, try to build the glucose, not try, to build the glucose molecules. So in this case, the glucose molecule, or what it will become, the glucose molecule, is going to be the one that the one that is reduced, and the things that bring things in are going to be the ones that are oxidized. So the exact opposite of what we saw in chapter 7, which should make sense, because chapter 7 is about breaking down the molecule, and this is about building the molecule. So all those words that we talked about in the past, exergonic, endergonic, potential, kinetic, all that stuff will come into play as we build. Again, I say we, as the plants build this molecule. But again, just talking about it, we can't say it enough times, so you can really encompass what's going on. Oh, I just realized, man, I didn't do my proper opening. All right, but as always, I got my glass here, and you're thinking, whoa, 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 whoa. Look at that clock. It's pre-lunch. What are you doing with the pop already? Yeah, I know. I wasn't very good today. As a matter of fact, I tried to really be bad, because I had to go to the grocery store first thing this morning. You know, as soon as it opened up at 8, because what we put in the smoker, I needed some wood chip. I wanted some special wood chips for a special flavor, or different than what I had, is what I'm trying to say. So I had to go to the store, and so I'm swinging back through, and I'm passing the pump, which is our local convenience store. We got 2. We got a Casey's here, and then we got a pump, which is a local one, and everybody tries to go to the local one, or at least the ones that at least I do anyway, support the local businesses, and try to get me a big old drink, 44-ounce drink, and their ice machine was down. Well, I didn't want just a cup of pop. I mean, I can just pour me a glass when I get home, so that's what I did. But yeah, so it's not been a traditional day. Oh, but enough about that. And I got my notes too, which you've already know because we already typed some up. So let's move on here. Let me close this down, and let me slide this over. Okay, and so what we're going to look here now, let me make this a little bit bigger. And yeah, don't look over here, because that's going to be a pretty cool metaphorical situation there, so don't pay no attention to that. But it is going to be a strong teaching room, no doubt about that. All right, so this is, as you can see, let me get my pen here. We are going to be doing an overview of the light dependent section, as you can see right here. And then we'll briefly talk about the bottom part here, but that's part 2 of the show. And we won't spend a lot of time on that just yet. But once we get there, we will spend plenty. All right, so what we're talking about here, just like over here, so we've got the sunlight. Okay, and this is going to be our first real we're going to start talking about what's going on, okay, or how it's happening. It's probably a better way of saying it. But you got your sunlight, okay. Now these, obviously, we're in the photosystem here, so we're in the chloroplast, and so we're talking about these, the granums of the thylakoids that we talked about yesterday. Okay, so the sunlight strikes, okay, this big stack of stuff of the thylakoids. Okay, and you can kind of see it right here, and here they are right here. So sunlight strikes them, okay, and it keeps, because the sunlight, never stops. So it just keeps coming, pulsing, pulsing, pulsing. And we collect, and eventually enough energy, okay, just like with baking a cake in the oven, you can't just turn it on any temperature and stick the thing in there and instantly it's done. It takes time. Okay, but over time we collect enough energy, okay, to get those electrons, and the term is excited or just energized is a better way of saying it. Sometimes in a high school setting, and you can understand this, you can't say the word excited. It just brings all kinds of different thoughts there. So energized is the word we try to use. That way it avoids all the snickers. So energized. And once that happens, okay, then some stuff really starts to happen. And we'll bring some diagrams up that really showcase that. But that's the idea. We're going to collect until we get enough energy to get this process going. Okay? And now when I say collect, let's think about it like this. Let me go on and come back to it. And then, of course, it's a pathway. Okay, we got sunlight, water, and we got the NADP, and we're going to end up with some oxygen. Okay, and when we show the simulation, you're really going to see where that oxygen comes from. But we kind of looked at it in the first because we talked about where the O's come from, where the H's come from, where the C's come from. But it's the idea. We get this reaction going. We get the electrons moving. And get the products made that are going to be used, the ATP I'm talking about, and the NADPH, and then we're going to bring in some CO2, and then we're going to get that glucose molecule made. So let me close this, and I think my slide is down here. Yes, it is. And then from here on in, we're going to be going just straight, okay, straight to the right, or it would be east if you were looking at a map. And there's just a long string of diagrams that we're going to be going through. But at my house, probably my favorite, well, it'd be a tie between the grill or the smoker. I love those things and love being around them, doing things with them. But if I had a second favorite tool, device, machine, whatever you want to call it, it would be the old satellite dish. Yeah, because that brings them some good things. So once the good food is made, then we find us a nice ball game. Like last night, well, we had two things last night. We finished up the Dodgers Braves, which was a great game. And of course, then for the evening, we had the incredible football game. I don't know if you watched the Georgia Alabama game, but that was a great game. And man, Alabama looks so good. We're definitely headed to another Alabama Clemson championship game. It sure looks like to me. But anyway, satellite dish brings that stuff to me. And I don't know if you know how a satellite dish works, but the function of the structure is pretty simple, and it's pretty easy to see that the satellite information comes in here and collects on this dish, and it just collects, and then it converts it. Okay, you can't see it on the back side, but it comes up into this reaction. We're going to call this the collection center right here. And then if you look at yours at your house, you'll see a bunch of wires and those what goes to your TV or to your box. It goes to a box anymore. So you can watch it, change the channel, and then boom, there it is. So we collect. So we got the collection center, and we're just going to call that CC. And then we got the reaction center, okay, which is where things get converted, okay, and then it's sent out into your house. Well, the same thing happens, or I shouldn't say the same thing, a similar thing is happening, okay, with photosynthesis and the chloroplast. Leave that one up here. Okay, we just talked about this being like the antenna or the collection area and the reaction center. And if we convert that to, oh, I don't have my pen up. If we convert that to what's going on to chloroplast, so this is that same membrane that we saw earlier, and this is that same blob, except on the other one it was blue. Why they made it blue, I don't know, but it should be green, chloroplast. Okay, so now we've got these pigment areas, and we talked about pigment in the first video. That's just things that can absorb the sunlight and convert it. So we got the light that strikes it. This is the collection center. So this would be the same as this. We're going to collect. So that's the old CC, the collection area. Okay. Then we collect, and when we get enough energy in there, okay, the energy travels to the reaction center. So this area is just like this right here. Okay, and when we get enough energy, these electrons get energized enough, okay, and they're grabbed by this electron acceptor, and then boom, they shoot out of there, okay, and start this process of headed towards the Calvin cycle. And this happens both in PS for photosystem 1 and 2. Okay, so this happens twice. Okay, it's like a long assembly line, and it happens in the first part, and it happens in the second part, and then it gets transferred over. But we will talk about all that. So now, as I scroll down here, okay, same picture. So again, the light strikes. Okay, that's the collection center. It's collecting all that energy. Okay, and when enough energy is collected, okay, it gets those electrons energized to where they jump to the acceptor and then are moved down the process. And you can kind of see this process right here. So, but guess who supplies? Guess who supplies the electrons? That's right, some water. Okay, you got that water. So we've got H2O. So we split that molecule, little hydrolysis going on there, split it. And then we've got the two H's. And we got that O. Okay, and this happens over and over and over and over again. So the H's they got the E's. Okay, they've got the E's. They supply those E's, and then we got the oxygen, and of course, the one oxygen combines with another oxygen, a little covalent bond there. So you get you some O2, and guess where that goes? That's right, it gets released out into the atmosphere, and it comes to us. So again, incredible situation. So you've got your light coming in, energy is collected. Okay, the hydrogens supply the electrons. When enough energy is collected, just like with the cake metaphor, when enough energy is collected by the cake, okay, all the materials form that cake product, and it's done. But just like here, enough energy is collected, the electrons are energized, and they jump to the acceptor, and then boom, they're out of there. And same thing over here. You can kind of see it right here. Okay, all this electron, and then when the electron gets energized enough, boom, it shoots out of there, gets accepted by the electron acceptor, okay, and then an ATP is made, but we'll talk about that. And then the electron acceptor comes back and gets another one, and then boom, does the whole thing again. So boom, just continually goes through that process, getting those electrons up and out and onto the next part of deal. Because this is a general deal, and so this could be one or it could be two, either one, but the same thing happens in both. What happens next is different between the two, but this same process of collecting energy, the electrons getting energized and coming out of that center, being accepted by electron carrier, that happens in both over and over and over again. Okay, so we will close that. And as you can see, I'm at 26 minutes. All right, so now what you saw here, you see right here. So, oh, didn't mean to do that. That was not a strong teacher move. Okay, so what we see here, we also see right there. Okay, now what we're going to talk about is what happens after the electron is accepted. Okay, so again, now we're going to label this. Let me go a little bit bigger here. You know what? I need to stay back here on 62 because that's the right number for me. And let's just make this a little bigger. No, that's not what I want. Alright, what's going on there? Let's do this. No, that ain't working either. Well, let's like it. Yeah, let's do that. Oh, now I got me a little. Now if I click on it, can't enlarge it. Well, and I got that little like sign on there. All right, so I guess we're just going bigger. So if you want to like the picture too, you can as well. All right, now that was not a strong teacher move. All that delay there. All right, so again, what's happened over here is happening here. We're collecting energy. Okay, collect. When you get enough, the electron, and now you can see the number 2 right there. And you may be saying, why is there 2? Because remember, there were 2 hydrogens. They're each giving up an electron. So we got the 2. And so the energized electrons eject out, and now they're accepted by an electron carrier here, just as we talked about over here. Yeah, and there's that picture. You don't want to cover up the picture. All right, and it travels to the next photosystem. Now, you may be wondering, why are they called photosystem 2, photosystem 1, or what you might be asking is, why is it photosystem 2, then photosystem 1? Why isn't it switched? And you know what? I have been answering that question with, I don't know, for a long time. You know, all these years of teaching. My only guess is that they figured one out first, and second, I don't know. But you would think it'd be 1, then 2, but it isn't. So just recognize what we got and move on. Or as my mom would say, just let it go. Okay? And she always did her eyebrows to it too. That was like, what you're doing is dumb. Let it go. So we got the electrons up and out of the photosystem 2, and now we're to photosystem 1. Again, we're collecting more, we're collecting more, we're collecting more. And then boom, okay, where those electrons are energized, again, up and out, we electron carrier, okay, and you can see that the NADP is reduced, and those electrons, those H's are now on this NADP, and now it's H, so it was reduced because it gained the electron. So that's the idea, and you can kind of see some end product stuff here. End products of photosystem 2 is the electrons and the oxygen. Remember, the oxygen gets released right here, and we got those electrons. and then the end product for NADP or photosystem 1 is this NADP, but it's got those H's on there, NADPH, and it's carrying those to the Calvin cycle. And when we look at the animation, we have some questions at the end that we're going to process through, and it's about some of this auxiliary information: who supplies the electrons, or where do they come from, where do how do we replace electrons? You can see like we've lost these. Okay, so where's the next set come from? And again, they're going to come from here, and then this carries it over here. Water supplies the electrons over here, and so, but we'll cover all that in time. Just make sure you're understanding the basics of collecting, okay, energized, releasing, electron carrier, transport, collecting, energized, electron transport, electron carrier, and then boom, then we're off to the Calvin cycle. From here you would go, you would go to the Calvin cycle. Oh, that's not a very good C right there. All right, so we will close out the ink tray, and now move over here. And this is the slide that we will actually start. We'll make sure we put some notes down so you can know what to write. And so we'll put text boxes underneath each the photosystem 2, photosystem 1, and we'll talk about all this down here. But man, look what we got right there in step 4. What does that look like? Where have you seen that before? Dramatic pause. Man, I hope you were screaming, hey, that's the APT syntax we saw in chapter 7, cellular respiration. Love that thing. Love that machine right there. And again, you can just look at the diagram. It's doing the same thing it did then as it's doing now. So, and you may be thinking, well, I thought cellular respiration was where the ATPs are made. It is, okay? But guess what? What we're going to be doing with that glucose molecule, it takes some energy. Okay, you can't make a sandwich without doing some work, without getting some work done. Okay, you can't make a glucose molecule without spending some energy. So what the plant's cells do, we need some energy. All right, let's make some. How efficient is that? How incredible is that the plant cell takes care of its own business? Okay, it needs some ATP to get this job done. Heck yeah, let's make some then. Oh, I don't know if I can say heck yeah. I don't know. I'll have to look that one up. But it makes it. But yeah, it's awesome. So let's get some notes in here. Let's close this down. Let's get a little text box here, and we'll put this bad boy underneath photosystem 2. And just for space, I will do that right there. Photosystem 2. All right, so we'll go through these real fast, and you may have already written all this stuff down. Just pause it and listen and wrote down what I said. But if not, this is our official note collection spot. Oh, see what I did there? Yeah, that's a strong teacher move, to tie in words that you're using into what you're saying. No, let's go sunlight. When enough is collected, I didn't mean to go this long, but it's the way it's working out. Energized. So I'll have to slide that over, or just what I know what I can do is I can do this. There we go. Okay, when enough is collected, electrons are energized. Electrons eject. electron carrier grabs, electrons, transport to PS1. Okay, so there we go right there. So that's part one. Now, just so you can see it, what's labeled step one here is this stuff, but the electron carrier part, that's officially step two. That's what's going on right here in this little transport train right here. So just recognize that. Okay, then we'll close that out and pull up another little box here and put that underneath photosystem 1. So we will go PS1, outline format, and again, same steps. Okay, same steps. Okay, and then NADPH, transport, and then we'll put it officially here, transports to the Calvin cycle. So again, photosystem 1 collects that sunlight, converts it to energy. When it gets enough energy, the electrons are energized, electrons eject, electron carrier grabs the electrons, and they transport it to the Calvin cycle. So, and that is, and if that wasn't enough, okay, now we got part 4 over here, okay, where all these hydrogens, okay, that we're collecting here and we're using the electrons with, pumping through there. And not only are they being part of adding to the NADPs, but they're also funneling through the ATP synthase and making some ADP. And you can already see from our next diagram the ATP right there. So it's amazing. Okay, amazing. And the cell, I just thought about saying this, the cell did all of this, and guess how much of its own energy it used? None. None. The sunlight supplied all of the energy. And we talked about that yesterday. That was our I shouldn't say yesterday, in the first video. That was our initial thought that we looked at and thought about, is the fact that this whole process, this amazing process, okay, the energy it supplies by an object millions and millions and millions miles away. How in the world you couldn't design a system to where you used energy, okay, from some place that supplies energy that is millions and millions, or just miles, hundreds of miles away, okay, hundreds of miles away, to process a machine, okay, in your shop. Like, for example, you couldn't, so let's do it this way. You couldn't design a system to where you used energy that was generated in St. Louis to run a machine in your house, office, shop, whatever you want to call. Couldn't do it. Okay, couldn't do it. The wiring alone, I mean, how you gonna run wire to get the electricity through? And even if, some people might say, well, what about those windmill farms or nuclear plant electrical parts? Okay, and then you can bring the energy to you. Maybe you could make that debate that it's supplied, but it isn't a direct connection. It's not directly from that source to you. It's going to go through some kind of plant, or that buys it, and then the energy, that electricity, is then pumped to you through their system. Okay, so it's not a direct, not like this. I mean, this is just how awesome this is. Oh, and I can talk about it for, I can talk about how great it is forever, let alone just talking about it in itself. All right, so as you can see here, we are fast approaching the end, and you're going, finally. And we've been going at it for 40 minutes, and we're just through the first part. Well, the second part's pretty quick, or certainly a lot quicker than all that stuff. So just a little transition here, and then we'll talk about this bad boy over here. And when you look at that, you're already thinking, dang, that is some complex stuff. That is kind of like the Calvin cycle and all that stuff that's going on. But the ATP that we just made over here, the ATP synthase, we're going to use that. Okay, and we're going to use some NADPHs for the hydrogens. Okay, we got some CO2. So just think, we got some C's. Okay, we got some O's. And we're going to drop off some H's. Okay, guess what we're going to be making? Yeah, that's right. We're going to make a little C6, H12, O6. And yeah, some little glucose. Okay, some glucose that your cells will use for all the energy that we need. So let's close this, and we'll bring this one just right up front and center here. And let's make this a little bit bigger. One way up to 8. Oh, that's right, because we had to get bigger there for the other one. All right, now just like with the Craig cycle and how it leads into, okay, it's a cycle. So the final product, or what it ends up being up here, okay, is the start of the whole process. So let's break this down. We've got this molecule right here. Don't I have a pen? Oh, I closed it to move it over. That's right. So we're going to start the conversation right here. RUBP. Okay, that molecule, and you can see the breakdown of it up here. Pretty complex. Got this long string. Got some C's in here. So you can already see that carbon backbone right here, bunch of OHs, and going back to past discussions, you see that OH on the end. What's the first thing you think of? Yeah, attachment points, connection points, okay, bonding sites. All right, but we're bringing in some CO2, okay, and it says carbon fixation, okay. I know the word fixation is an odd word in a science setting because it means so much different in these day and times with all this like stuff on TV and the hoarders and whatever. And my precious little Avery, she can get that way. And I've said this story many times, she's a pit bull lab mix. She got the lab body, the pit bull head, and the pit bull head is what drives the rest of the body. So when she wants something, okay, we are going to do everything we can to get that. So if we're playing like tug of war, or she's got a ball in her mouth and I'm trying to get it out, and that's our game, or if I have the bone that she's chewing on and she wants it, man, she is all focused on that thing. And if it's no longer playing a game, then you've got it to really just get her attention. And I mean, you have to, say, hey, no, we're not playing no more. We're putting it up. We're done now. And even then, she may sit beside the basket that it's in, knowing that she can't get in and get it, but she really wants it. So not that fixation. What we're talking about here is modifying it so it can be used. Okay, modification so it can be used. So you can see here that we got the CO2 molecule, and when we get over to here, okay, you don't see CO2 anywhere. That's because it's been slightly separated now, so it's right up here. Okay, they just wrote it a little differently, but that's where it's at. Okay, so it's attached to this ending molecule, and now we've got ribisco. Okay, or it's called ribisco. It's the name of it, and you can see they've added that CO at the back. Okay, just to reference that we're adding that carbon. So again, when we say fixation, what we really mean is that we are attaching it, and again, it's here, we are attaching it to the starting molecule. Okay, and so now we go from this, okay, and now everything gets kind of moved around, and we've got this. So now we're here. Okay, so our next part, okay, we're going to have a reduction. PGA, okay, is reduced to GA3P, and again, that's a lot of letters there, but just hang with us. Okay, here's your PGA right up here. Okay, and that's just the name of. Okay, so it goes from this to this, and now it's called that right there. Okay, our next step is looky here. Okay, all those ATPs we made back there at that what would be up here at that ATP synthase. Okay, we're using 6 of them right here, and like I told you, hey, it costs some energy to get this made, but we made them. Okay, with energy from the sun. So we're taking those ATP and we're reducing, we're taking those phosphates off. We're getting some energy here. Okay, and that's the reduction. So when we say reduce, we're reducing the molecule. Okay, so we're adding things to it. Okay, it's getting electrons, so its energy level is coming down. And so if the molecule is being reduced, okay, the ATP molecules are being oxidized because they're losing the electrons. So we want to have that reference to redox that we spent a bunch of time in 6 and 7 talking about. So the glucose molecule is being reduced. Yeah, and the ATPs are being oxidized. Okay, then the same thing here. So it's happening here, and it's happening here. Okay, the NADPHs are losing the electrons, the H's, and the molecule is collecting them. So the molecule gets reduced, the electron carriers are getting oxidized. So what we're really doing here is we're collecting a bunch of H's. Okay, because we can get, like for example, we got 3 CO2s, so that's the 6 oxygen we need right there. Actually, that's the 6, and we got 3 of the carbons. Okay, but we already had a good carbon backbone here, so we just need to make sure we have a bunch of H's, and they're showing up right here. And now we've got, okay, this conversion, and now we have, okay, the GA3P, which is the official end product of the Calvin cycle. And then the GA3P then is combined, because you can see one of these molecules makes half of a glucose molecule. In other words, it makes C3H6O3, and you just got to add 2 of them together. Okay, you just have to have one more. That would be half. 2 halves is, equal 1. And you can see we got the 3 carbons. Okay, and the 1, 2, 3, 4. See, 2, 3, 4, 5. Where's mine? Where would it? Am I missing the 6th one? Is it on this? Yeah, I bet it's on this because it's what came over. So it's probably, it's in here attached to that somewhere. So they're just not showing it. That would be where the 6th one is. And then we got the 3 oxygens, the 1, 2, 3. So we've got half, as it says right here. So this would be just this half right here. And then we'll go through, so it means we got to go through the Calvin cycle twice to get a glucose molecule. Okay, and then once that splits off, okay, what's rest? It's converted back to the RUBP. Okay, and then we use 3 more ATPs, and then it gets us back up to where we were, and then right back into another cycle. So I felt the sneeze coming on there, and I'm going to try to get away, but luckily it didn't happen. We don't want to see that. So that is the Calvin cycle, okay, in its situation there. So that whole process, again, amazing, okay, amazing stuff. But just recognize that one time through makes half the molecule, and you got to go through a second time to get it complete. But your plants are doing this all day long, just boom, boom, Calvin cycle, Calvin cycle, Calvin cycle, Calvin cycle, cranking out as much glucose as it has the components for, which is why I need to take in the oxygen, or I need to take in the CO2, okay, take in the waters. And when we look at this animation, which we're going to now, you will see that. So let's get a quick drink. And let's pull that up here, and let's adjust actually I need to go full screen first, and then we'll adjust that. Yeah, let's move myself just over there a little bit, and let's get myself a little draw thing here. And we want green, obviously, and let me see what this is going to look like. Oh, it goes away. So I'm going to have to draw me an arrow, and then you just have to recognize that it's there. And then it looks like it's just going to go away. So it's not going to stay on there very long. All right, so, and here's the thing I got to click through. We're going to go through the first parts real quick. Okay, so what we're talking about here is we got thylakoids, the granums, the stromas, okay, and then that's what this little diagram here is what this is. This is the stroma part, and this is the thylakoid space. So inside, okay, inside of those is where this part is, and in the darker green, the neon green is the thylakoids, the darker green is the stromas, just so you can know exactly where we are, and this would be that membrane. All right, so again, inside the thylakoids, the membrane, and the stroma. So we are on one of these thylakoid membranes here. All right, this is all of our players. Got the PS2, okay, the PS1, and of course, then the Calvin cycle is somewhere over there. All right. Okay, so there's our sunlight that we're going to collect, and we're going to collect, and that's a little photon, and it's going to travel down to the electrons, and they're going to collect it, okay, and we keep collecting, and then boom, okay, starting to get some energy going here, and then one more time, and then boom, now they're energized, okay, and they're getting ready to eject. Okay, so there's the ejection, as we've seen, in all those diagrams that we talked about. Nothing new going on. Okay, guess what's going to happen next? Okay, they travel. Okay, it's part of a question later on, but what they're not showing you is that it gets there with the electron carrier, NADP, and we put those H's on there. So it travels down through, and this is that. If you think back to the diagram, we had the photosystem 1, and we had that like protein looking thing that traveled through to get to the second part. That's what this is. And this right here represents the ATP synthase. That's just not a good diagram of it. So it's traveling through that system. Okay. And now all these H's show up. Okay. And right now they're not showing you where those came from, but we know that, and it's one of the questions that they're going to ask us. Okay, and so here's that ATP synthase that we're making. Now we're making some ATP. There that is. Okay, and now we're to PS1. Okay, again, we got our electrons down here. We're collecting sunlight, collecting sunlight, collecting sunlight, and then boom, energized, eject, and now we're going to travel down through this process. Okay, and now we're to this, and the next move would be to go to the Calvin cycle. And now we're going to go through a series of just kind of follow-up questions and stuff that the person who made this animation wants to ask and to clarify. Okay, so here we go. Problem 1. Who is the final electron acceptor for the ETC in photosystem 1? Over here, in other words, who's going to take these electrons to the Calvin cycle? That's what that's asking us. Okay, how can we replace the electrons lost in PS1? In other words, we don't have any electrons right here. Where are they going to come from? Okay, question 3 or problem 3. Who is the final electron acceptor for the electron transport chain in PS2? In other words, who's going to take these over to here? And 4, how can we replace the electrons lost in PS2? In other words, where are these going to come from? And we know where they're going to come from. Those bad boys. But we'll get there. All right. So question 1. Who's the final electron acceptor for the ETC in photosystem 1? In other words, who's going to take these guys to the Calvin cycle? And you should know the answer to this. That's right. The NAD. Okay, NAD. Now you're asking, okay, where's the P at? It's already given it up for the phosphate. And now it's going to take this NADH. Okay. So it's going to carry those that way. Now the next question, how can we replace the electrons lost in photosystem 1? In other words, where are these coming from? And you should know the answer to that. Oh, big freeze right there. Big freeze right there. So now my communication is probably going to be off from the lips moving, but that's all right. Maybe time to be thinking about a new computer, I guess. But the question is the same. And here they go. Okay, so they're going to replace that. Okay, that's right, from the ETC in photosystem 2. And now our next question. Who's the final electron acceptor for the ETC in PS2? In other words, who brought them over to here? And again, we know who that is. Okay, PS2, PS1. Oh, acceptor. Yeah. I was thinking transport for some reason. And lastly, how are we going to replace the electrons lost in photosystem 2? In other words, how are we going to replace those electrons up there? And we saw this, we've already talked about this way back at the beginning. And so this should not be a spoiler alert to know that it's the water. Okay, the water's going to come in. Okay, you've got your two H's, you got your O's. We're going to split that. Okay, that's where all these H's came from. And then, there we go. We're taking those H's and those electrons, and they're going to go down here. And that bad boy right there is going to be released out into the atmosphere. And there you go. And there you can see that the O2 is truly releasing. That's right, snagging them from the water. And again, the beauty of this situation is all these H's again are going to be pumping through here to making those ATP. Don't forget about that. And so what we have here is this is being released. And then these bad boys are headed to the Calvin cycle, as you can see there. All right, and that's our animation right there. So we don't want to, let's close that. We don't want to end on in this area. Oh, I guess I just got to open it up down here. There we go. Oh, done. All right. So, right at an hour too. Good timing right there. Hey, so I hope you enjoyed that. Hope that was beneficial. Hope you got a lot out of that, and that you are deepened. Your knowledge of the photosynthetic process is much deeper now, and I enjoy talking about this stuff. I say that way too often, but I do love this stuff. Love you guys. Appreciate you guys. And hey, with that, let's say goodbye. Let's get out of here until next time.